and welcome to another episode of SCFG Live, Science Club for Girls Live. I'm Hannah with Science Club for Girls, and that was Mr. Music with your theme song. Very nice. It's great to have you back in the studio today, Dr. Music. Why not? Mr. Music, Dr. Music. Very nice. Well, I'm so glad everyone could join us again for another episode of Science Club for Girls Live. I'm excited for today's episode because it's going to continue to build on to what we talked about last week with cells. But first, we got some pretty big news. What's that big news, you ask? Well, it's brought to you by our fabulous geologist, Kate Hardock. Let's take a look.
Hey, didn't see you there. I'm Kate. Y'all may know me as Kate the Geologist, but I also mentor with Science Club for Girls at the King Open School, and I'm on the board of directors and head of the development committee. I'm here to tell you about an exciting announcement from SCFG. In less than 2 weeks, Science Club will be hosting our Catalyst Awards, and this is our 25th anniversary of the Catalyst Awards and our first ever virtual Catalyst. So the awards are on Thursday, October 1st, and starting at 6:30 p.m. It's going to be so fun. We're going to share some of our videos from in-person programming and some of our virtual programming as well. Also, we have some great guests to look forward to, including Kaylin and Ashley, some of our SCFG mentors. Please register to attend the event at tinyurl.com/virtual catalyst 2020. No spaces. Well, I gotta go lick some rocks. See you later.
Coming up on October 1st, our Science Club for Girls Catalyst Awards. It's going to be a fantastic night, and it's totally free to join us. So go ahead and log on to that website that is being shown on your screen right now. I'll link it in the chat below as well. And register. We can't wait to see you there. All right, now time to talk about what we talked about last week. Cells. Last week we talked about cells, and we said that cells, kind of like Legos, right? Legos are used like building blocks to build things like houses and cars and castles and whatever else you want to build with Legos. Cells are the building blocks of all living things. Like this puppy. This puppy is made up of trillions of cells. It's a multicellular organism, just like us humans. But remember, this ice cream? No, not made of cells, because it's not a living thing. Now, we also took a closer look inside of an animal cell by making a model out of pizza, which I love, and I hope you love too. And we looked at all of these teeny tiny organelles inside of animal cells. And each of these organelles plays a really important role that allows the cell to work properly. Like the ribosomes help to build the proteins that the cell needs. And the nucleus, well, the nucleus is super, super important because it's the control center of the cell. In fact, it tells the cell what to do and how to function. And that's all because of something called DNA, deoxyribonucleic acid. I'll say it again because that's a long word. Deoxyribonucleic acid. Tell that to your parents or friends or family and they'll be really impressed. Okay, so what is this DNA? Well, this DNA is kind of like a recipe. It tells the organism how to function and how to look. I remember when I was younger, I would ask my parents, why do I have blue eyes? And they would often say, well, it's because of your DNA. And that's true. DNA is what tells humans what eye color to have, and it tells the lungs how to function properly. So today, we're going to take a closer look at this really important thing called DNA. Now, to do so, we're going to take a look at a model. And that model is made out of candy! Like my second favorite thing to pizza. So I thought we have to make it out of candy. Now, this model is a really blown up model of your DNA because your DNA is about 200,000 times smaller than this. We really couldn't be able to see a single strand of DNA without a really powerful microscope, so we've blown it up to make it candy size. Okay, now what you notice is the DNA kind of looks like a ladder. If it's in your body, the DNA actually looks something like this. This is called a double helix structure, which is oftentimes what scientists call it. And the reason why it's wrapped up like this is because your DNA is really, 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 really long. And so in order to fit inside of your nuclei and your cells, it has to kind of roll up like this to shorten. All right, let's unwrap it for now to take a closer look. What do you notice? Well, Well, I noticed that there's 4 different colored gumdrops. And these gumdrops represent 4 chemicals that help to encode information and tell the cell what to do and how to function. And these 4 chemicals are adenine, guanine, thymine, and cytosine. And these 4 chemicals bond together in pairs to form the rungs on our DNA ladder. Now, what's cool about these chemicals is that they always pair in the same way. Adenine always pairs with thymine, and cytosine always pairs with guanine. Cytosine and guanine. So you'll always see yellow and red, that adenine and thymine, and orange and purple together, that guanine and cytosine. Really cool. Now, there are about over 3 billion pairs of chemicals in your DNA. So again, this is only a small section of that DNA. Really cool. All right, now let's talk about why scientists study DNA. Well, there are a couple of reasons why scientists might want to take a look at DNA and extract it or take it out of the cell. Sometimes they use DNA to form new medicines, and sometimes they'll use DNA to genetically modify or change vegetables to maybe make them resistant to insects, and other cool things like that. So this is what's called DNA extraction, and we're actually going to get a chance to do that today by using strawberries. Now, why are we using strawberries? Because strawberries actually have a lot of DNA inside of them. In fact, about 8 times more DNA than a human does. So we're going to get a chance to look at that today. So in order to do our DNA extraction, we'll need a few things. The first are strawberries. In fact, you'll need 3 strawberries in a Ziploc bag. You'll want to make sure to cut off the stems before putting them in the Ziploc bag. All right, now it's time to make our extraction liquid. For that, you'll need a bowl, 1/3 cup of water, some salt. You'll actually need a 1/2 teaspoon of salt. So let me get my teaspoon. Cool. You'll also need some detergent. You can just use dish detergent, like what you'd wash your dishes with. And you'll need a tablespoon of that. Cool. Once that's all in there, go ahead and give it a mix with a spoon. All right. This is what's going to allow the cells in the strawberry to pop open and take that DNA out so we can take a look at it. All right, open up your bag of strawberries, and you'll want to put 3 tablespoons of your extraction liquid in the bag. 1, 2, and 3. Zip it up nice and tight and squeeze out all the air. All right, now for the fun part. You'll want to mash your strawberries into mush. This should take about 2 minutes to do that. Make sure you're mixing all those strawberries and the extraction liquid together. Now, because this takes 2 minutes, I already did one ahead of time. Once it's all done, it'll probably look something like this, like strawberry pulp. It's kind of gross. All right, next you'll want to strain your strawberry pulp into a small clear glass jar. To make sure it doesn't spill everywhere, I would recommend using a funnel. Funnel, strainer, and then our strawberry pulp. Gross, gross, gross. All right, now to help push it through the strainer, you might want to use a spoon. Mix that in, push it down, and you'll slowly start to see it going into your jar. Set that aside. I'm just going to use my bowl so it doesn't get all messy. And the next thing is you'll want to add 1/3 cup of rubbing alcohol. Now, because this is a chemical, I'm going to make sure I am extra safe and put on my super safety goggles. Remember, when you're using chemicals, it's a good idea to have something on your eyes, whether that's sunglasses or goggles, just to make sure the chemical doesn't get inside your eyes. Not good. All right, so rubbing alcohol, you're going to measure out 1/3 of a cup of rubbing alcohol. Oops, some strawberries got in there, but that's okay. All right, 1/3 of a cup. And now for the fun part. You're going to carefully layer your rubbing alcohol on top of your strawberry mixture. You want to make sure that the rubbing alcohol and the strawberry mixture don't mix together. Instead, this will just create a thin layer on top. So kind of hold your glass at an angle and slowly pour on the rubbing alcohol. All right, this is what will eventually help to really pull that DNA out and separate it from the rest of our strawberry mix. All right, so give that a little bit of time and you should start to see some clumps coming together. Ooh, I'm starting to see it already. Now, in order to extract your DNA, you might want to use a spoon or some tweezers. In my case, I think I'm going to use a spoon. All right, I'm going to hold it up close to the camera so you can take a look. You'll start to see that white stuff that's on top of the strawberry mixture, like in between the alcohol and the strawberry mixture, and that's DNA clumped together. That's crazy. Oh my gosh. Now, if you want to try and pull it out again, you can use kind of like a spoon or tweezers, or you can just kind of take a look at it in the jar. That's crazy. So what exactly is happening here? Well, our extraction mixture, especially the detergent, is causing the cells to kind of pop open, releasing that nucleus and releasing the DNA. Now, the salt is really important too, because the salt is allowing the DNA to clump together. We wouldn't be able to see a single strand of strawberry DNA because it's so small. We need a really powerful microscope. But when they clump together, we can see them because they create a larger body. Oh my gosh, look at it now. That's crazy, all that DNA. Now, I challenge you to try this at home with so many other different fruits. Try kiwis, try bananas, using the same extraction technique. Pretty cool. Be sure to post photos about it and tag SCFG Live. All right, well, that was really fun. We got to see what a DNA model looks like, and we got to see some actual DNA. Remember, DNA is really, really important to allowing us to look and act and function the way we do. Now, I had a friend, Julie, who actually got a chance to sit down with a scientist who uses DNA in her work. Let's go ahead and take a look at their discussion.
First of all, thank you for agreeing to this interview. So could you please introduce yourself?
So I am Professor Amy Yee. I am a professor of, my department is Developmental, Molecular, and Cellular Biology at Tufts University School of Medicine. I have a PhD in biochemistry, and I grew up in California and went through all the California public schools and was a undergraduate alumni of UC Berkeley and UC Davis.
All right, thank you. Can you tell us a bit more about what a typical day is like as a researcher and a professor?
Pretty much it comes down to two big responsibilities. One is I do a fair amount of teaching at all levels. So I do teaching, and then I also do research, and embedded in that research is also teaching the students to do to learn how to do research. So the research now comes in many flavors. I do biomedical research. So we use the computer a lot to look at databases on behaviors of disease and manifestations of and how disease shows up in patients. But we also do experiments in the lab, at the bench, trying to make new discoveries that will eventually help. We hope will eventually help patients. In fact, that is my primary goal with my career and my life's work. Is although I function at the level of the science and elaborating and discovering those basic science principles. What I want to do is to package this into a way to make an impact in patients' lives.
All right, thank you. So how did you really start becoming interested in biology? Where did this all start?
So in school from the very beginning, I was always interested in math. And, as you go on from second grade, third grade on, is first grade, maybe even now, is that I was always interested in the science. So I carried that throughout school. When I was an undergraduate at Berkeley, which is a major research university, I learned that you could be a scientist and you could study science. So even before that, all I wanted to do was to go to medical school because I just loved biological sciences. But as an undergraduate, I learned and experienced that I could be a scientist and I could be in a position to make those discoveries that make a difference in people's lives. And actually, as we see now, is that the science has a major impact in society. We see this with the COVID-19, is you do have to listen to the science. And actually, I think in this current situation, I appreciate more the fact that at Berkeley, and even before that, I have a relatively broad education. It's good to be interested in everything. Because as a scientist, we need to not only do the science, but we need to be able to write about it and we need to communicate it. So some of those things are taught in classes that you don't expect to be related to science, say the history, the political science, the literature. So it's good to be, I'd say as a scientist and a professor, it's important to be a good student in lots of things, of course, emphasizing what you love. So what I'd say to all the younger students is be interested and curious in everything and follow your passion. So I was very lucky in that I was inspired. I always had the passion for science, but I was really inspired by some great teachers that allowed me to explore my passions.
If you could give one piece of encompassing advice for the young viewers out there, what would it be?
Be the best student that you possibly can be, and be curious and follow your passions.
All right. Thank you so much for this interview.
Wow, that was awesome. I loved how she talked about being curious and how many different parts of her education ended up being related to science. I also love the fact that she was talking about how much she loves that science is there to help other people, and that's like her overall goal. So, awesome work. Well, that's all for our show today. I do want to remind everyone about our upcoming Catalyst event on October 1st. We actually are going to be taking a break from our shows just for the next 2 weeks as we get started and get ready for our fall programming, which is coming up. So I'm really excited about that. So be sure to check out Catalyst. Otherwise, we'll see you on October 9th. And this has been another episode of SCFG Live! Science Club for Girls Live. Oh, one more look at the DNA. Pretty cool. Bye, everyone.